Right now.
No, he's still like.
Are you redoing your explanation?
Yep. For money. No, that's a K-bar. Sweet.
Can we do this, like copy this pretty much?
Do what?
Like, can we copy like this idea?
You can.
Like with the magnets and stuff?
You can. I'm gonna kill Billy. Oh, don't tape that part.
Please kill Billy.
Don't tape that part.
Hey, how do you know what goes in that little ball? Yeah.
This?
Yeah.
That's just anything you want to make.
It's just random.
Believe.
Okay.
Yep.
Okay, go. You ready?
Where you want me to start?
Yeah, like DNA model.
Okay. My DNA model is made up of a series of nucleotides or base pairs. I have 15 base pairs in my model. And each nucleotide has a phosphate component, a sugar component, and a nitrogen base component. Adenine, thymine, guanine, and cytosine. And they're held together in the middle by a hydrogen bond. And the molecule actually twists like a spiral staircase, forming the double helix shape. The genetic code of the molecule is the sequence of the bases. That controls all cell activities and passes on genetic traits to other cells and to offspring. DNA replicates by the DNA helicase unzipping the hydrogen bonds completely, and then new free-floating nucleotides come in on both sides of the rails and find their complementary nitrogen base, A to T, C to G, and fill in both rails all the way down until we have two complete copies of DNA, in theory, side by side, that are put into the new cells during the cell cycle during mitosis and cytokinesis, because every cell needs DNA or it can't function. Just double checking if I missed anything for the C level. Nope, that's the C level right there. Shall we go on to the B?
Yes.
Okay. Transcription, like replication, happens in the nucleus. It's when DNA makes RNA, and it's a similar process. We have new enzymes called transcription factors and RNA polymerase that control the process, but the DNA unzips again. This time only a gene unzips, and RNA nucleotides come in one at a time to find their complement on one side, the length of the gene. I pre-made this piece to slide in here to show you how that works. And once that gene has sequenced a piece of RNA and put it together, the RNA leaves the nucleus and the DNA or the gene goes back together. There are three differences between RNA and DNA. It's a single strand of nucleotides. It has a different base. Uracil replaces thymine. And it has a different sugar, ribose sugar instead of deoxyribose sugar. And there's three kinds made: messenger RNA, transfer RNA, and ribosomal RNA. And all the RNAs work together to put together a protein. So that is the B level. And a word I might have not used was expression. When the gene turns on and makes RNA, we say a gene is expressing itself. You want the last piece too? Okay. Translation is the last piece of my model. It's when the RNAs have left and gone out to the ribosome area in the cytoplasm of the cell to make a protein. So it happens in the cytoplasm. The three kinds of RNA work together: ribosomal RNA, messenger RNA, transfer RNA, to put amino acids together to make a protein. The piece of messenger RNA that gets expressed is called an exon because all the non-coding pieces were removed. Those were called introns. So the messenger RNA goes out to the ribosome. This is ribosomal RNA. And the first codon, the first triplet of bases, is read by ribosomal RNA. And that calls for a certain amino acid to be brought to the ribosome by transfer RNA. And the way we get the right amino acid in the right sequence is transfer RNA has an anticodon on it, and messenger RNA has the codon that uses RNA rules to temporarily bond adenine to uracil and cytosine to guanine, A to U, C to G, as my legend indicates on my color code. And the first one is in place, and that transfer RNA sits there, but the ribosome moves to the next codon, which is going to be lysine. When lysine transfer RNA gets here, this transfer RNA lets go and leaves, but before it does, it transfers the amino acid it had to the piece that came in, amino acid that came in. And this goes away until the ribosome slides down to the next codon, which is AAA. This calls for proline to be delivered by transfer RNA. When proline gets here, these amino acids let go and attach to proline, and this transfer RNA leaves. And then the ribosome moves down again and calls for arginine. Sorry. Arginine comes down next. When this transfer RNA gets here, these amino acids transfer to the arginine, and this leaves. And we've got one more left. The ribosome reaches the last codon. Stop codon comes into place. The amino acids clip onto it, and arginine transfer RNA leaves. The stop RNA then also stops the making of the protein and releases it, just the amino acids, in the right sequence to go out and do its job inside the cell. All chemical reactions in our body require enzymes to make them happen. So proteins are hugely important for us every day we're alive. All this RNA can be recycled and used again, and more protein can be made. A future application of this kind of knowledge is we can make man-made proteins now inside of bacteria by putting human genes inside of bacteria and produce drugs for people who are sick, like insulin, for example. We're done.